interesting interesting structures. And if you're looking at the Sirius cam in the overhead looking down at D2, you can kind of see that some sediment has been deposited in these sawtooth type of fractures.
Okay, Dan.
And you can see that see what we're talking about here with these fractures that are propagating down to the south.
These are just not going to be doable. And there's an abundance of this type of coral, and I think moving on, Sirius is pretty close to the ridge right now. Okay. So if we can saddle you port and get up north of Sirius right now. Okay. I'll settle port. You can see the ridges off to his left here. Yep. That, I think, will be in much more in reach than any of this.
While we have a little time, I also do want to give a shout out to the NOAA Deep Sea Coral Research and Technology Program. This program was really instrumental in bringing the Okeanos as well as other NOAA resources to the region. So the NOAA Deep Sea Coral Research and Technology Program has, over the 3-year cycles, focused on different regions of the United States and the deep water coral and sponge resources in these places. Done very important work in the Gulf and in the North Atlantic, Alaska, and now the next 3 years doing work here in the U.S. Pacific, and this is part of some of their efforts.
Sirius is settling on that now, and we're just going to have to
carry
forward.
And then that's also been the Pacific Island
Deep
Sea Coral Reef Initiative.
On your using your sonar, line up your head on the local high.
Watch lead, this is pilot.
And pilot will sidle left and line up. And I'll fill in watch lead while you run.
Okay, if you want to talk to him.
Yeah. Okay, watch lead. Nav, sorry. You're muted, I think. I can't hear you. Turn down? No?
I don't hear him either.
There you go. So Sirius has decided what it wants to do. The ship, we're moving the ship the best we can, but the current swung us full circle around the ship as the current switched and has gone upridge of us. So we're going to race out up ahead of Sirius and hope we can find a fan up here. It looks like they're pretty abundant, so we're continuing uphill, and I think hopefully the current, we're now in the current on the ridge, and if we stick to the ridge, that should stay consistent. Hopefully we can make moves, and Sirius will agree with us. We certainly don't want to be stretched out too much. It's not good for the tether, and makes it dangerous and things like that. So we're just going to push up here and hopefully find a similar species for us to grab. No, you're doing great. Yep.
Yeah, so we were just having a discussion here with our navigator and our pilot. So yeah, it's a challenging environment here. So current switching and lining all the vehicles up for those folks who don't understand. So it's basically it's a 3-vehicle team. So you have the ship tethered to the Sirius, which is a smaller underwater research underwater vehicle that's tethered to the ship, and that hovers about 100 feet off the bottom. And then tethered to that vehicle is the Deep Discoverer, and that's the images you see of the bottom. And so when you're moving around objects, it takes a lot of coordination to move the 3 vehicles in tandem. So typically the ship moves, then Sirius moves, and then the Deep Discoverer moves. It's kind of like a caterpillar, so one moves, then the other one, then the other. And so we had a particularly challenging terrain there, and the currents whistling around, so we're just going to move around it, and it looks like we'll find that same primnoid coral later. We did see a whole bunch of them there, and at least a few at the beginning, so I'm quite confident that we'll find another one of them that it's easier to grab and not perched on top of a pinnacle where the microcurrents make it even harder to get. So right now in view is a chrysogorgid coral. That's another family. So I was talking about
Hi, can we get a ship move, please?
the primnoid coral that we imaged earlier that we're trying to collect.
Do 30
This is a
meters
other family that are
Yeah,
also
co-pilot, you're
Gorgonian
tilt up.
corals.
at 2 tenths.
Gorgonians are a group of octocorals, so octocorals coming from 8. So those tentacles and the polyps there, you can see that there's 8 of them. And you can actually also see that the tentacles themselves have tiny branches. And so that's characteristic of the octocorals. That makes them different from the hexacorals, which, for example, the scleractinian corals, the ones that you see on shallow water reefs, belong to. Black corals are also hexacorals. Anemones are hexacorals. So in those corals, you have polyps with tentacles in a multiple of 6 that are unbranched.
Co-pilot.
So as soon as you see those branches on the tentacles, gives you an indicator that this is an octocoral.
Where Sirius was.
And then within the octocorals, you have different groups. You have the true soft corals,
We'll make sure that
or
Sirius
alcyonations. Those have tiny calcite spicules that are fused, so those are skeleton, but they don't have a hard axis or this dark kind of skeleton in the middle.
Co-pilot.
So
Sorry, I think
these
you
are
need to speak up.
Gorgonians, so they have this axis. So if you were to remain all the tissue from there, you would have the axis like
There you go.
a dark skeleton
She's
that
just
made
trying
up.
to get a shot of the full thing.
So that makes these different. For example, the mushroom corals that we saw, a whole bunch of them, those are true soft corals or alcyonations. So if you were to remove the tissue, all you would be left with microscopic tiny calcite spicules. But they don't have a true skeleton or axis. And so yeah, that's a beautiful view. And this is another Gorgonian coral, the reddish, pinkish individual next to it, and that's a bubblegum coral. That's in the family Paragorgidae, and this is the genus Paragorgia. And the common name of bubblegum coral comes from when these actually retract their tentacles, you'll see a tiny, minute bumps on the skeleton. Bubblegum corals are also interesting. There's a whole bunch of species of these, and mostly restricted to very deep waters. There's one species in
Camera back.
particular. It's found off the waters of New Zealand, and there's some individuals of that species have been measured to be over 5 meters tall.
Ship's only moved.
So yeah, some of these animals in the deep sea are impressive in terms of their size. And as was pointed out by a few of our shore-based scientists, also not just their size but their age. You have some of these corals and sponges that can attain ages of multiple centuries, and in some cases several millennia.
No, it seems peaceful here.
It's peaceful for where you are.
So
Sirius is
those
the same
bottle-brushed
experience.
type colonies, there's one to the bottom of our screen and one to the center of the screen. That is another octocoral and another Gorgonian octocoral in the family Chrysogorgia, Chrysogorgidae, and that's going to be Chrysogorgia geniculata.
Fully stretched out.
And next to it is a
because of the
tiny
way Sirius
glass
swung,
sponge, and that's going to be the genus
we made it
Walteria.
move closer to D2, but in fact the whole time you're
And
moving farther and
yeah, it's
farther away.
quite impressive. This
Deep,
the
as I told
dense
him
and
that you were
diverse
moving
community
closer,
that
so
we've
he's
seen
looking at staying
throughout
where he was, thinking he's
the
fighting
dive
current,
today.
He's fighting the tether. So that's why you're heading.
So I just want to point out these long continuing fractures that are running parallel to each other and along the axis of the ridge. So this is some of the fractures I was just talking about a little bit ago, where they are propagating away from the head wall and from our right to left, and these blocks are becoming separated at these long parallel fractures and moving down dip direction. Or what I mean by down dip is the lava flows that form these layers are actually dipping in a certain direction, in the direction that they flowed downhill. And downhill in this case is to our left, which is more or less south right now. The ROV is kind of looking just about 30 degrees west of north. And so what we're seeing is these orientations are more or less
Can you zoom out?
Yep.
north
Thanks.
or more northwest west, and the dip is going down to the southwest. And so these blocks are moving due to the weakening of the fabric and the lava flows that have made up this ridge and the landslide that slid off to the right. And so we're going to keep our eye out for any more of these parallel fractures along the ridge line as we keep on going up, because as we keep on going up, the head wall that results from the large submarine landslide only gets steeper and steeper and becomes a larger feature. So we'll keep an eye out for more regional type of geology as we ascend up this ridge.
Yeah, there are a couple questions about our plans for the rest of the expedition. So tomorrow, unfortunately, is going to be our last dive day, and we're going to be diving on another seamount that's about 60 miles south of here. That's unnamed, still unmapped seamount. So we'll be after today's dive heading south and then mapping that feature and hopefully find some attractive targets to dive. And then, unfortunately, we have to transit straight to Kwajalein Atoll. That's where this cruise will end next week, Friday. And it's such an enormous distance. We have close to 1,500 miles ahead of us in transit. So there will not be any dives in the mid-pack range. We had to make this decision because actually the weather in that area is right now worse. So throughout the course of this expedition, we had some major storms in the area, some enormous storms just north of Midway that prevented us from going towards the very northern portions of the Papahanaumokuakea National Monument. And now finally those storms have cleared, and we have a nice weather window to about tomorrow. And yeah, so right now those storms have passed, and in some cases the seas are even rougher south of us. So we made the decision to make the best use of this narrow weather window and stay in this area and get all the dive days in. So there we have another chrysogorgid coral, several of them actually. So those kind of bottle brush shaped, and then one next to it is another one of these bubblegum or paragorgia corals.
So I think we're about to do a shift change, watch lead.
Copy that.
But I think conditions are settling out, so
Excellent,
be
thanks.
in a good position to move up the ridge.
Excellent. So yeah, we're just going through a change in personnel in our piloting team. It's lunchtime, and some of our pilots are rotating in and out, so everybody has a chance to grab lunch. And then once we're all switched over, we'll continue around this ridge. But fortunately, our pilots have done a great job at navigating across this pinnacle, and where we have some confused current flows. And so as soon as we have the personnel change complete, we'll continue transit of this ridge.
Does it seem like things are swinging the right way?
We are currently at a depth of 1,994 meters.
Okay.
So we have approximately 3.5 hours remaining on this dive, and we do want to cover some ground, so we'll try to go up this ridge, increasing the pace a little bit, hoping to see not just characterize this community in terms of what species it might contain, which of course for that we need some samples and some images, but also how large this community extends. So for that, of course, we need to cover some ground and see if we perhaps might pick up the end of it, or if this is actually a continuous feature that goes along this ridge. So this ridge actually goes to almost the summit of this underwater mountain or seamount named Castellano Seamount that up until yesterday was not mapped for more than half of the feature. And as a interesting side conversation about where are all the fish. So far we are
still changing
a single fish on this dive. And this appears to be a common theme, that in the places where you see a lot of the corals, a lot of the sponges, a lot of the habitat forming animals, you don't really see the fish. And then in some of the more sediment patches where you have less corals and sponges, you'll start seeing more of the fishes appear.
Hi, Daniel.
Yeah.
Andrea, it's kind of an interesting observation for this part of the world, because other areas we've been to in the Western Atlantic, there could be several species of deep water corals, and we see a lot of different species of fish in high abundance, particularly in the canyons off the northeast U.S. Atlantic that the Okeanos visited in 2013 and 2014. Even in some of the similar depth ranges. I mean, this could be related to depth too. We're a little bit deeper. Yeah, I don't know.
Yeah, thanks. That was Andrea Quattrina from USGS, who has been a science lead on a lot of these cruises aboard the Okeanos Explorer, and has a lot of experience diving in different environments and a lot of these deep water environments.
Yeah. Roger, thank you.
And so that's
I always like these little scale worms.
those are chitons.
I have those in my yard.
They
had some
very
time while we were
less
doing.
depth.
yeah, that's a good find. And yeah,
While we were doing the pilot change here, so
these are
nice
very
texture on it,
unusual. These plated.
That's a
It's
full zoom.
a group of mollusks. So they're characterized by these 8 plates. So they there's actually a whole suite of chitons, different species of chitons, also some shallow water ones.
Much else we can do there.
Yeah,
so.
that's.
And in fact, there's a few
Just thinking
species in the Caribbean that are found in
Hey,
very
there we go.
shallow
That's your
waters.
second
They're also
Okay.
interested and used for as food.
Coming out in 3.
So yeah, they're characterized by those 8 plates. So that's a type of mollusk.
All right, I got a co-pilot and almost a nav soon, so we'll move on.
And Bruce Mundy from Pacific Islands Fisheries Science Center just offered a great remark here about when we're talking about the productivity and lower biomasses of fish, perhaps. And so we are in the Central Pacific, where we have a little bit lower productivity than other places. So this is this particular gyre.
So I'm told we're just sitting still right now.
But yeah, and that's something that we're hoping
3:30,
to start answering as the Okeanos Explorer goes to further places that are spaced over large gradients, is to try to figure out, what are the patterns across entire ocean basins.
330.
In the past, a lot of this deep sea research had focused
Copy.
perhaps on single seamounts or single sites. And some of
Pilot's
the
ready for a move. once
some
you
of the
guys
early observations of
figure it
those deep water explorations was that pretty much every seamount ended up being completely different, fauna, and there was.
Co-pilot, did you get you got the scoop from Carl, so we're going to try to air to the southwest side of this ridge.
Okay.
If you want to.
Sorry, I just had a quick pause there, talk with Nav. But so yeah, the early explorations of some of these seamounts was that they were coming back with completely new faunas, and a lot of these seamounts and There was a lot of talk about every seamount being
Just saying
completely
if you needed
unique
to
and
close the
endemic.
cap
And,
sorry, cap the lens
or-
or anything you can.
All the fauna was endemic, and as we're going to more and more of these seamounts, we start seeing that that was in fact a problem because we had low sample numbers. As we go to more of these seamounts, we start seeing that in some cases there are similar faunas,
Let's make it.
and in fact in even similar
Let's make it
faunas
315.
across different oceans. And so there are connections between some of these deep water ecosystems that are widely spaced apart. And so that's something that's a wonderful
We'll do
thing
30
by the
meters
Okeanos
start.
Explorer is that
That's okay.
it's covering over the next 3 years enormous distances diving throughout the Pacific, and then it's done work in the past in other ocean basins. So it's really by doing these kinds of surveys in different parts of
Bridge,
the
yeah.
world that we start
coming back towards
getting
the
a first
co-pilot
glimpse at
before I start
biogeographical
getting
patterns.
tugged.
How are the faunas different?
Like a ship move
So for terrestrial and shallow water ecology, and we scientists have defined kind of biogeographical provinces, so distinct parts of the world where you have kind of more similar faunas and floras, and that's not really been done that much in the deep sea because we have very limited observations, and hopefully as we continue exploring these deep water realms, we'll be able to see more of the connections and how some of these places might be more similar or more different to each other.
Degrees, but I can't. Auto heading isn't functioning, and it doesn't appear to be a tether.
It's just being blown. You're
Yeah.
just being blown. Rotated a little, I believe.
Current depth
Thank you,
is
Bridge.
1,990 meters. To our left is a very
So we just
conspicuous
started
looking
that move
coral.
If you want to get him back to 315.
That's Iridogorgia magnispiralis. Individuals of that very same species were measured to be over 5 meters tall, about the size of an adult giraffe. And currently those measurements of that coral that were just taken a few years ago up in the Papahanaumokuakea Marine National Monument by Dr. Les Watling and his colleagues. Those measurements are still the largest Gorgonian coral in the world. And so yeah, we see several primnoid corals still there in view. That fan to the right, that's another primnoid coral. All those unbranched corals that you see in the background is wire-like looking. Those are all bamboo corals, unbranched bamboo corals. And yeah, very interesting that we've not only captured during this dive a dense assemblage, but also a fairly diverse assemblage. And can we take a quick look at that coral to our lower left?
We definitely can. The yellow one?
Yep.
All right. Yeah, we're waiting on a move here from Sirius, so we can...
Copy that.
Ooh, that's kind of tall there on port.
Yeah. Good view of them on the minute cams. Go ahead, video.
Yeah, coming in.
Center it up.
Holding.
Yeah, that looks like a dead piece.
Go ahead.
So yeah, that's
that full?
It's a
Let's
fragment.
just go full. I don't think it's too much.
So yeah, that's a dead piece. So this is a fragment that either
What
the
is it
branch
that's
broke
driving
off
that move? Like 2 minutes?
or a colony toppled over. So that coral is now dead, and you do not see any polyps on it anymore. And so this is the exposed skeleton that I was talking about earlier. And there's a little shrimp hiding underneath. There's also a barnacle on one of the lower branches that just we had in view earlier. Yeah, very.
Is that a snail on the bottom?
Oh
left?
yeah, that's a gastropod. Yeah, good eye.
All right, video. I think we probably have
Yeah,
this.
feels like it. It's not going anywhere. We're wide.
Not sure they messed with the Z bias after they picked up their rock.
So for
Seemed pretty
those
heavy.
folks who might have been
Oh, I
part
just
of our
lowered it
descent to the water
down to there.
column, we had a very interesting conversation. We actually had the captain of the Hokulea
Rock's big
call
enough
in,
that the lid can't close.
and we had not a nice conversation. So the Hokulea is part of the the Polynesian Voyaging Society that is currently in its effort over several years to
Still pretty far
navigate across all the world's oceans, and using
Roger
traditional
Just
wayfinding
taking
and
this
traditional
opportunity
ways
to
of voyaging. And we had a nice lengthy
tune
discussions
the
about
vehicle.
some of the similarities and differences between our ongoing efforts here
Feel pretty good about
using
it. Alright.
more
See what's out there.
modern ways, but in comparing that to traditional wayfinding, and but also seeing many of the similarities that, and in particular, that the overall goal of these missions is really the same, and
Look at
it's
these
about
big ones.
raising awareness, about educating the
These
public
may be the same that
about
they
some of the.
Wanted to sample.
Yeah, Bobby, I think so. And so, yeah, raising awareness about all the wonderful things that are down here and all around the oceans.
Well, the ship move was only 30 meters, so if you want to
Yeah, so
settle in for a sample, you got plenty of time.
yeah, that guy is the same thing.
Yeah, I'm still waiting for
Should you- Are you going to want a sample of this?
Yeah, I think the consensus is to grab that. So if you set up, I think that's. Yeah, so let's see, 2 large colonies here, and there's a few more in the back, and this is this very large primnoid coral that we think is in the genus Paracalyptrophora, and has a great interest by several of our shore-based scientists to get a sample of this very large fan.
Actually, do you see any snippets on here that are more interesting than others?
I think just one of the distal branches that you can grab, kind of, without.
Do we want it below a certain branching point, or
Yeah, so maybe that branch on the right, maybe halfway up, the very righter on the right side, most.
yeah, we can probably.
Maybe halfway up, just.
Video, let's get a quick image.
Roger, coming in. Trying to check my focus to the left.
So yeah, we're imaging now a primnoid coral that we believe is in the genus Paracalyptrophora, one or more dominant and abundant species here
Coming out a little bit.
in this community we have imaged so far, and we're going to go ahead and collect a sample.
Tilt down a little bit.
There's great interest by several of our shore-based scientists.
It's nice. Coming out a little bit more.
Yeah, I think that's good. I just wanted to get a quick image before we. Full wide?
Current depth
Full
is
wide.
1,988
Go
meters.
pilot, you want to ramp up hydraulics for me, please?
I'll keep an eye on the
Can we get a quick laser on maybe?
Coming on.
Thank you. That's good.
Should I stow? Retract the pins and
Well,
close the drawer?
let's see. How are we doing on HPU? Oh, yep, we're up. We're spun up.
Yep.
You shouldn't have to do anything with the drawers right now. You may not be able to get them open since we're on a slope.
Copy.
So let me just reach out and snip first. Alright, hydraulics coming on.
So yeah, we see several commensals also on this large
If you're
fan.
comfortable, co-pilot, let's bring
So on the very left, there's a crinoid, and that's a feather star.
Mini Zeus up on your screen.
Those
please.
are another group of invertebrates that is in the same group as you have urchins, sea
I can
stars,
get that for you if you want, co-pilot.
as well as brittle stars, which are throughout the colony more towards the right. Those are
And there.
ophiurids. And it's something that we see continuously, is that a lot of other invertebrates using these tall colonies as habitat.
Just come over, get
And
the
as-
coral and my arm.
How's that for you, pilot?
Yeah, that's good.
Gonna take your sonar, co-pilot.
Copy.
Can you come right, actually, some more, please?
Going right. How's that?
Looks good.
Just looking ahead, today's dive is gonna. We have about 3 hours and 15 minutes remaining on the bottom. Gonna be leaving approximately at 3:30 Hawaii time.
Arm
And
feels
then
so
we
much
do have a
bigger
dive
now.
tomorrow on a seamount that is about
actually
60
next to
to
it.
70 miles south of us. And that's going to be unfortunately
we'll just come up
the
from the
last dive of this expedition. But the Okeanos Explorer is going to continue working in the region,
Make sure we keep an
doing,
eye on Sirius.
Copy.
both mapping and remotely operated vehicle operations, both in the Marianas Trench Marine National Monument and Pacific Remote Islands Marine National Monument. So yeah, here in the Pacific, we have some of the largest marine protected areas on the
Watch lead
planet.
down here. Look like a good branching point.
And the Okeanos Explorer is going to a lot of these over the course of 3 years to try to understand some of the
I can try and
unique
work it out.
fauna of the region.
Watch lead pilot.
Go ahead.
Yeah, does that look like a general good area right there? I think there's some branching. Can you go partial, Zeus, please?
Roger. There?
Yeah, it looks like there's some branching right above there. All right, we're going to cut it off. Let's come wide, please.
Full wide.
Co-pilot, can you come up on Sirius, maybe 3 meters?
Copy.
Okay, I'm going to hold that there.
Very exciting moment. We just collected
gonna
the
come
first
off just a little
biological
bit.
specimen of this dive. This is a primnoid coral that appears to be.
This is our first sample, I believe.
This is our second, so this is going to be Spec 02
Sorry,
bio.
first bio.
Yep.
So we should be able to just go into the port J box. Or sorry, port bio box. I'm going to come up. All right, I'm going to hold right here.
Okay, and to review the procedure, close
Stand by
drawer,
1 second.
copy.
Or Nav can walk you through it, but I need to
So now,
settle this first.
so close drawer, retract port bio, and then extend the drawer.
Close the drawer, retract the port bio,
Yep.
extend the port bio, and then extend the drawer.
Copy.
All right, I'm clear of the drawers if you want to start opening.
Okay, so I'm retracting port bio. I'm closing the drawer. That puts me back in stow.
Retract both of them, yep.
Oh, okay.
So retract port bio
Yep.
and
Retract
starboard bio.
starboard bio.
That just ensures the pins are up top.
Yeah.
Copy. Close the drawer again. All right,
Since we
now
don't have another sample.
extending port bio,
It's good practice.
and now I'm opening the drawer.
Yep.
All right, Pilot, I'm gonna. I guess we'll leave the mini Zeus in place. Just trying to get a good view.
You can move the mini Zeus.
All right.
Yeah, that's where he's going anyway.
All right. So I'm opening the drawer.
Watch lead, I have Spec 02 bio.
That is correct.
Thank you.
Minip cam show we're good.
You can open up faster than that. Yeah, you're clear.
Copy.
So yeah, I mentioned that our
Let's get a quick
Our
check
di-
on Sirius. It looks like I'm
our
above
crews
you. You happy
here
with
and
that,
this
Nav?
these
Or
surveys
you
as
can
part of
watch it?
this expedition are
Speed drifting. Sonar.
about to end, but
Yeah, I've got.
there's several
If
other
you need to switch the mini Zeus
expeditions
camera for a
to the
second, you can.
Papahanaumokuakea happening later this year.
Running up on that monitor.
There is
Just briefly.
several cruises to go
we're safe.
explore some of
Okay.
the
14-meter delta.
deeper
over 15 meters
photosynthetic
off the
reefs,
deck.
There you go.
diving, and
Just get out.
200 to 300 foot range. It was a
All right,
month-long
yep, we're clear to
expedition.
switch.
There's also another expedition going to some of the shallower reefs, as well as another expedition that's going to be focusing on intertidal communities. So the Papahanaumokuakea Marine National Monument really has a really diverse group of different habitats here. You have terrestrial habitats and some of the emergent land features, and then to the very deep sea. And so there's several cruises to try to pick and get a glimpse at some of these different habitats and different environments. And of course, the technologies are very different depending on what depth range you're looking at. When you go into the deep sea, of course, you need submersibles and remotely operated vehicles. Some of the shallower reefs, scuba diving is and then on the islands themselves. So that's something that's an ongoing effort, is to try to characterize the very wide diversity of different habitats, species, environments we have in these places, and that's really another value of some of these large-scale marine protected areas that we have in the Pacific, is that they protect a whole different group of different habitats that are diverse. And then they also protect entire ecosystems. So you might have an incomplete ecosystem all encapsulated within a protected area.
Okay, I'm going to just let it sink in there because I can't really get much farther in.
Copy. Standing by to close the drawer once it gets in there.
Nice.
Stand by. just try to push this little piece in.
Yeah, so after we are all done installing
Is it going?
the sample,
Just try and get these pieces out of there so we can get a good seal. Okay. Go.
Nice.
well done.
All right, now we can close the drawer. Everything should get a good seal now. Those last little pieces.
All right, closing drawer.
When the drawers close, I think we need to do a vehicle check on the configuration. So this has drifted 30 meters since we thought it settled.
And if we do have a second, there's also a quick request to do a snap zoom on that stump that's just to the right of the coral we sampled, that white kind of speck there, about a meter right of the colony.
And I've swung my heading around as well. Almost due north. Was it 315?
All right, let's bring our normal screens up.
Should I spin down the HPU pilot?
Yep, let's spin it down. Come out of my positions.
Before you go anywhere, let's make sure we're happy. You can see the way that the tethers.
When we get a chance, pilots, the swing arms look like they're really just giving us a really hot spot directly in front of us. It'd be nice to diffuse that a little bit.
Okay.
We can take a look at that one thing they wanted to look at first if you wanted.
You need me just to push forward right now.
Yeah.
Oh, wait. Ooh. Sorry, Watchley, we're coming back. What was?
No, that's quite all right. We'll see another one of that.
Are you sure?
Yeah.
Sorry.
That's quite all right.
I was trying to get in position. Completely forgot about the request.
On the towards the back there, we have our first what we've been referring to Isodella species lyrate, very conspicuous, kind of candelabrum-shaped octocoral, and that's another one of these that are in a revision. Those names are in the process of being changed. That's a bamboo coral.
All right, Nav, where do you want me? Or where do you and co-pilot want me? You want me to come back?
You're kind of far out in front.
All right.
If we're going to do...
Go ahead, video. Let's partial.
Roger, coming in partial. There's a fish down below you to the right, too.
And pilot, if we're going to adjust the...
Wait,
Lisa,
what are we
I've got a
going
great-
for, that guy?
Yeah.
Yeah.
Sorry, I
Fine.
should have spoken a little bit more.
Yeah, we see a
If we're gonna adjust
jellyfish
the port
there.
swing arm, now is a good time to do it. I have a
Yeah, go
nice
ahead.
view.
Yeah, it's a hydromedusa.
He's inside.
Yeah.
So
We-
those are actually
Either one's
interesting
anyway.
that they are in the same kind of overall
More
group,
exotic
and the
looking
corals
ones.
you see
There's that fish
they
down there, too.
both have
Yeah, let's snap.
stinging cells. Or
I've
nematocysts.
seen these already.
And there we go, our very first.
Hey, Daniel, this is Andrea. I was just going to say it looks like our first fish of the day.
First
Yeah.
fish. Disregard.
And so that's a rat tail or a macrurid.
We're just going to get an ID on this one video. I got to move back
no
to
worries.
my co-pilot.
So yeah, the rat tails
I think we know what it
in
is.
reference to that
Okay.
long tail, and
Coming
in
out.
McCrur it actually means long tail.
Coming back to you, co-pilot.
Copy. Is that any better?
Yeah, that could be a Kumba species, but hopefully Bruce Mundy can
Blowing out
join in and tell us a little bit more.
I know. Maybe once we got a...
Yeah, I was just going to guess Coryphaenoides longicirus, actually, but I stepped out again for a moment, and then, of course, the fish comes around, so I just caught a glimpse of it at the end.
Yeah, I had a hard time seeing a long chin barbel.
Yeah, okay, I didn't, it was turned away, so I didn't see that. I just saw it looked like it had extensions on the pelvic fin.
If that's our move,
I see.
yeah, let's get on 315.
And then beyond that, like I said, unfortunately, I stepped out for just a moment again when the fish came around.
Looks like it's pretty hot in front of Sirius right now.
Yeah, some of these macrarids are really hard to identify from video alone. That's for sure.
We want to go up there.
Yeah.
We want to keep it to our starboard then.
So to
Yeah, and unfortunately, I don't think Alan Andrews is here now, so he's not going to be able to provide screen captures like he did yesterday, so we'll have to wait until something's posted on the FTP site.
340, you happy with
So
that?
yeah, we see
Sure.
a lot of these unbranched bamboo corals here in this assemblage. A couple of sponges there, that cactus-like
Some already
looking
there.
animal, that's a
Yep. coming in
Walteria
this review.
sponge, and then we see a very large pink coral, and that's in the genus Hemicorallium.
Let's do 330.
And right at its base are several mushroom corals.
Swinging left to 330.
And so yeah, those mushroom corals are what are known as the true soft corals. They don't have a skeletal axis,
Ship move, please.
so you don't have a
Range 30
axial
meters.
skeleton
Bearing
running around the
330
middle, and
degrees.
that's what you would see in the
Speed
Gorgonian
0
corals.
decimal
So,
knots.
for example, that pink coral. Then next to that, there's a bubblegum coral. So yeah, there's
Good copy.
4 different corals right on that rock. There's a bubblegum coral, the red one, a pink coral, a chrysogorgic coral, and a mushroom or anthomastus. Perhaps we can take a quick snap zoom on one of these unbranched. They seem to be so abundant right here.
Got it, Watchley.
Yeah, one's right in front of you now on the right with the darker behind them are probably better candidates.
You had the grouping right here above
Yeah,
the
those
lasers.
3.
Yeah,
Read my mind.
any one
Yep,
of them,
right
yeah.
there.
Your F flat is going again, pilot.
Gosh.
Thank you, Bridge.
Yeah, and a nice density of them, all the same species apparently, and I don't know if this is a sea pen or it's
go ahead.
Coming in.
a Gorgonian. And yeah, it is a Gorgonian, and I believe this is Bathygorgia.
Start on this first one.
That's full zoom, by the way. Are you feeling that much current from port?
Yeah, there's
That thing looks like it's getting blown around.
enough to hold to bother the heading. Not really that much to.
Is it? Maybe actually.
Move this
Let's
around.
take a look.
Doesn't
Yeah.
really look like it's getting pushed too
Look,
much.
if we look at the
So
base,
keep it
perhaps.
tight.
Yeah. Scott just pointed out that this is a sea pen. Yeah. Yeah. Excellent. Yeah, so I was wrong. Yeah, it's Halopteris. Yeah, thanks, Scott. Yeah, we'd seen a different group earlier, a rock pen, an Antoptylum, and this is a
Scroll up. Look at the other one. Second in line. I think he's going to be left. There he is.
It appears to be very abundant here. As we zoom out, there's several dozen of them in our vicinity. Current depth is 1,978 meters. And the remaining about 3 hours we have on the dive. We are hoping to make our way up this ridge as far up as we can, just to try to get a sense for how far this community might extend. But we've both the amount of animals we've seen and the need, of course, to get good images of these.
Racking your focus.
Yeah, I was just going to say.
And then also a little bit of challenging current conditions is not really or slowed down our progress of this ridge, and we'll try to, over the remainder of the dive, cover some more distance.
All right.
All right,
Happy
coming
video.
out.
And there you get a nice appreciation for how dense this community of sea pens is right here. And Tina Molodtsova from the PP Shirshov Institute of Oceanology in Russia pointed out that all these sea pens, she honed in on the fact that they were not bamboo corals by the fact that they're too flexible. So yeah, the skeleton of bamboo corals is typically a bit more rigid.
Okay.
You're talking about the uppers?
The uppers.
You can try pushing them down, Andy. I think it should be down.
Okay.
But I think that's as high up as they go, looking out in front of us.
So they rotate though, don't they?
Yeah, but they have to rotate through so much when they're usually stowed. They have to face out forward. That I think that's all.
Okay.
The forward we can get on them
Yeah,
right
yeah.
now. Did you try pushing them up?
No.
Yeah, perhaps.
To do that?
Yeah.
We can take a quick snap zoom on this sponge in front of us.
Push it down.
The other
I
up.
told you the wrong direction.
Gotcha.
There you go.
So that's as far as it goes.
Yeah, which is about where it was.
Yeah. They're want to look at that sponge too when we're done here.
Yep. Let's go look at the sponge.
I assume the other one's starboard's fully swung. I'll just adjust him real quick. Yeah. Yeah, they're both as
And they're
far
all the way out as well, or?
they could come out some. Looks to me.
Yeah. I'd be interested to see how that helps. We might want to do it once we're off bottom, though. It's a little hard to tell when we're this close, when our altitude's this low.
Copy.
So
Watchley, this was the sponge you wanted to see?
that's correct. And I believe this is going to be in the family Euplectellidae.
All right, go ahead, video.
Coming in.
And so I believe this is in the subfamily Corbitellinae.
Is the darker side better for...
It's actually not blowing out. It's just bouncing our light back. Oh, yeah. I mean, you get a little more texture in there. I'm not going to brighten that up because if we tilt up, we'll blow the whole thing out. Let me come out just a little
So
bit.
sponges are a very diverse group, and especially here in the deep sea, there's a bewildering diversity of different forms, shapes.
Let me split the two. There we go.
That's nice. Coming out in 3.
Onward.
Yeah, so far we really haven't seen that many sponges. A few of these polyopogon, and but yeah, this is more of a coral-dominated assemblage.
Yeah.
Conspicuously absent
I don't
in this
think we're gonna
coral
get stuff any
assemblage
better
are the
than
black
how we have
corals.
it. They're already pointing forward some.
Typically,
Right
they
from
are
the
quite
port, you can
abundant
see.
at some of these places.
this
We
mixture of
haven't
very
seen a
bright
single one
sediment
today,
or
and yesterday also we did not
material,
see a single
along
one.
with the dark rocks just creates a
So
lot
yeah,
of
this
piping.
seems
You
to
can
be
see that they're cheated forward a
Yeah.
little bit.
Yeah, that's
We don't
what I
really
was-
want to take them much more.
Yeah, a
Okay.
bit more
All right. Well, I'm gonna spin down HPU.
Yeah,
guys.
let's
the
bring it
primnoid
offline.
corals and pink corals and bubblegum corals seem to be the predominant, as well as the sea pens here in this particular area. For a few of our educators, and I know several classes are following us today and have been following us, I do want to point you to a wonderful collection of education resources that are a part of the expedition website. So there's a whole bunch of activities and lesson plans available. So if you go to oceanexplorer.noaa.gov, there's a whole education module there, and wonderful way of learning more about some of these deep water ecosystems, and in particular for educators, how to integrate some of these things into your classroom.
Is the ship still moving, or do we want to keep it going?
Negative. Stop. Oh no, it's got 2 more meters.
So in front of us, that's another pink coral.
We can keep going if you guys.
Yeah, let's
And,
keep
like
the
that.
ship moving. It doesn't sound like there's much interest in this area.
Yeah,
Okay.
yeah. We'll try to cover a little ground if we can.
Do you have a feeling for the ridge?
It looks like the
popping up?
substrate is changing a little bit here, Jonathan.
No. I think you're just going to have to
Yeah, it's mostly just been
estimate on that pack best you can.
volcanic rubble and hardpan.
340.
However, it looks like we're getting
I mean, it looks like there's some off.
some
Yeah,
of
it
the
looks-
in-place
I'd
lava
say
flows back in
330,
view again.
330
Yeah,
again?
I can
So
put
we
my
have
hand around.
a
Sounds good. I'll extend 30 meters.
approximate heading of
330.
about 30 degrees west of north.
Bridge, Nav.
These look like they're dipping directly into
Could we
us, so
extend
they're
30 meters
they're
to this
dipping
current
more or
move,
less south to southwest, and we're going to be still keeping an eye out for any of those fractures that are parallel to the
Good
ridge
copy, thank you,
axis,
Rich.
kind of indicating any kind of backward propagation of any kind of weaknesses
Okay,
that have been generated due to the landsliding along this ridge
Yeah.
line, because the ridge line is actually the lower part of a landslide scarp, or the cliffed out part that has been
Ridge,
oversteepened
open mic?
due to the removal of the material on the east and northeast side of the ridge by the landslide. So haven't been seeing much evidence of it for the past little bit, but it's just been mostly manganese, hardpan, and rubble. So see what we see as we keep on heading up.
Yeah, and it definitely does seem to be a very dense and heavy crust on this substrate.
Yeah, very dense, heavy crust. It's very old seamount, so it's Cretaceous, 80 million, 85 million years old. It's a lot different than the one that we saw yesterday of similar, presumably similar age, in that the seamount yesterday seemed to have had
Thank you, Bridge.
more accelerated erosion, and we might even have been able to see that if we were on the other side of the ridge, kind of looking at the headwall scarp where it's been excavated by the landslide. We might be able to see some of the edge on lava flow layering and things like that. But as we go up this ridge, we're kind of riding up and on top of what the surface of the lava flows would be.
Yeah, and then also very, very light
See
or to
anything
no
video?
sediment here.
No,
Yeah.
I assume we've seen
Not
all
a lot of
these,
sediment at all.
corals and
Again,
occasional sponge
different
before.
from
Don't hear them.
the dive yesterday, so just goes to show
We also
how
have a better monitor, so
some
you can see
seamounts
Yeah,
can
keep scanning.
be seemingly similar on the multi-beam maps that we use to help plan our dives, but radically different in their
It'd be
biological
really hard to
communities
call
and
out
geological
small stuff
landscapes.
like
Yeah,
that black and white checker.
and what makes it so exciting is that it
Yeah,
seems
sea
like every
star,
single
might be a
time we
dead stalk
go
up
on these
there on the
dives,
left.
is
Or a sea star.
you
Sea
see
star.
something
Is
new.
that sea star?
Looks like it.
Yeah, let's take a look at that sea star.
Am I going to be able to make it, co-pilot?
Just about.
Yeah, let's just go from here.
Yeah, it seems like it's... Yeah, I think I just got tugged.
All right, go ahead, video.
Roger, going in.